going to start off this video by talking about the cell cycle. G1 is the first part of interphase, and it is when the cell grows. The second part is synthesis, and it's when the DNA replicates. G2 is the last part of interphase, and it's when the cell gets ready for mitosis. Mitosis is cell division of nuclear material. Cytokinesis is the second part.
Cytokinesis is when cytoplasm splits or divides.
Early prophase is part of mitosis, and it's when the nuclear membrane starts to disintegrate, and then the nucleolus disappears. And the chromosomes start to condense instead of being soft and stringy like they would have been. And then the spindle apparatus begins to form between the poles. The next part of mitosis is late prophase. And prophase is split into two parts because it's the longest part of mitosis. Late prophase is when the centrioles appear and it sends out the spindle apparatuses to attach to the centromeres, and it starts to align them in the middle to prepare for metaphase.
The next part of mitosis is metaphase, and it's when the chromosome attaches to the spindle apparatus and they line up in the middle or along the equator.
The next part of mitosis is anaphase, and anaphase is when the microtubules shorten and it starts moving the chromosomes to the opposite poles of the cell.
This is the next phase is telophase. It's when the chromosomes are now at the poles of the cells. The nuclear membrane reappears and the nucleolus is formed and the chromosomes decondense and the cell starts to split.
Since we were on the topic of mitosis, we can go ahead and talk about cancer. Cancer is uncontrolled division of cells and growth of cells. Cancer is bad because it's when the cell just grows so much that it takes over all the good cells, and cancer is caused by carcinogens. In cancer, cyclins and CDKs don't work or function right in the cell cycle, so it causes the cancer to form more. Some possible causes of cancer are carcinogens, which are cancer-causing agents. You can also get cancer from UV radiation from the sun, and that would be skin cancer. You can get skin cancer from staying out and tanning too long or not wearing sunscreen.
Next, we're going to start talking about meiosis.
Meiosis means gametes.
Gametes are sex cells with half the number of chromosomes, which are haploids. The first step is interphase. In interphase, chromosomes replicate and chromatin condenses. Okay, in meiosis, there's PMAT1 and PMAT2. During PMAT1, the first phase is prophase 1. In prophase 1, the chromosomes pair, and it's called homologous chromosomes. And then they cross over, and this provides genetic variation. The nuclear membrane starts to break down, and the spindles start to form. Metaphase 1. In metaphase 1, homologous chromosomes align on the equator of the cell and they attach to the spindle fibers. During anaphase 1, the homologous chromosomes split and they start moving towards the opposite poles of the cell. Good job. After anaphase is telophase 1. The spindles break down and the chromosomes uncoil and they form two nuclei, and then the cell starts to divide. Next is prophase 2. Chromosomes condense and the spindles start to form, and the spindle fibers attach to the chromosomes. The next phase of meiosis is metaphase 2. During metaphase 2, both cells, the centromeres of the chromosomes line up randomly at the equator of each cell. Next, we have anaphase 2. In anaphase 2, the centromeres split and the sister chromatids separate and move towards the opposite poles.
Go.
Okay. In telophase 2, 4 nuclei form around the chromosomes, the spindles break down, and the cells divide into 4 different cells. Now we're going to talk about meiosis and its relationship with genetics. Meiosis forms gametes, and gametes are haploid sex cells. During meiosis, in prophase 1,
crossing over happens, and that is what forms genetic diversity in humans. Oh my God.
Next, I'm going to talk about non-disjunction. Non-disjunction is an error that occurs during cell division, specifically in anaphase. When the chromosomes split, it can cause there to be too many on one and then not enough in the other. What can happen is syndromes such as Turner syndrome, Down syndrome, Klinefelter syndrome, and even infertility.
The main challenge of this legacy cycle was to determine whether we thought that mitosis or meiosis was more important, but we believe that they are equally important.
Without mitosis, we could not have replacement of our cells, so if we had an injury, our skin cells could not replace. And without meiosis, we could not reproduce